into it. Okay, so what we're talking about, the topic is enzyme kinetics, and basically how fast is an enzyme making product at any particular substrate concentration. So this is the Michaelis-Menten formula. It's important to note the formula comes from the measured data. The data do not come from the formula. Okay? The data fit the formula, and I'll illustrate that here with an example of that we saw a moment ago, hexokinase. So the hexokinase enzyme, that's catalyzing the hydrolysis of ATP and the addition of a phosphate onto glucose to make glucose 6-phosphate. Okay, so if we want to describe the kinetic parameters of hexokinase and measure what its Vmax and Km are, we would do an experiment as shown here where we have equal amounts of hexokinase in each reaction, equal amounts of one substrate, ATP, and then varying amounts of the other substrate, glucose. Okay, and you would have a control that would have 0 glucose, I suppose, too. So we would set these reactions up, cap the tube and start the reaction, and then the measurement is the rate at which glucose 6-phosphate is produced. And the units of that rate would be something like moles of glucose 6-phosphate per second. Okay, so let's do that reaction and plot the data then. And the data we're plotting are the substrate concentrations. This is what we're varying in our experiment, and then the measured variable is the enzyme velocity. Okay, so at low glucose concentration, you get low velocity, and then as you add more glucose, the velocity increases, and for enzymes that are monomeric enzymes like hexokinases, the relationship between the substrate concentration and the initial reaction velocity is going to form the shape of a hyperbola. And by math, you can take those data points and derive that Michaelis-Menten formula from it. Okay. Now, in order to calculate the actual Vmax, we have to extrapolate this data to infinity. Okay, and we can't have a substrate concentration that isn't infinite, and we probably wouldn't want to. But if we did, we can do this mathematically, and the infinite substrate concentration, that's going to represent, or the velocity at infinite substrate concentration, that's going to be represented by the asymptote that the hyperbola approaches as the substrate concentration approaches infinity. So that is the Vmax, or maximum velocity, and this is this is going to be an intrinsic property of an enzyme. Okay. So Vmax, the formal definition of Vmax is the enzyme velocity that you would get at a hypothetical infinite concentration of substrate. Okay. So another intrinsic property of an enzyme is its Michaelis constant, or Km, and the Km is defined as the substrate concentration, so that's measured on the x-axis, the substrate concentration at which you would get 1/2 of the maximum velocity. Okay, so those two parameters are going to show essentially how steep this linear phase is of the hyperbola and when it kind of plateaus out. Okay, and that's shown here from your Marx textbook if you wanted to look at their version of it. So Michaelis-Menten kinetics, like the delta G0 prime from the first hour, these are, Michaelis-Menten kinetics are measured under very contrived conditions, right? You only have, substrates and enzyme around and nothing else. And that's not the way reactions happen in the body. However, Michaelis-Menten conditions Michaelis-Menten data are very useful for our understanding of reactions in the body. They can be used to measure different physiological or environmental conditions such as pH or temperature or what have you on the activity of an enzyme. Okay. They can also be used to determine whether endogenous substances within our body or different drugs that are made by a company, for example, are going to inhibit enzymes and how the mechanism by which they're inhibited. Michaelis-Menten kinetics can also be used to compare different isoforms of enzymes. For example, now hexokinase, that is the enzyme that makes glucose 6-phosphate in the liver. Hexokinase is the enzyme that makes glucose 6-phosphate in all of our tissues except the liver. In the liver, there's a different enzyme called glucokinase that catalyzes the same reaction. So glucokinase, that, we'll cover this. You're going to get sick of glucokinase soon, I'm sure, but that has a higher Km for the substrate glucose than does hexokinase. Okay, and those different kinetic relationships between the two isoforms, hexokinase and glucokinase, that's key for the liver's role in maintaining intertissue glucose homeostasis. Okay. All right, so just an example, these are measured under contrived conditions, but they have some very practical uses. Okay. I'm going to just skip over these, because I kind of forgot that I had them. Okay, so, and I think we covered them earlier, so you're not missing anything. So let's look at an example of how Michaelis-Menten kinetics could be used to measure the effects of pH. And so shown here. Now, pH is, you could vary the pH, such that you're completely denaturing an enzyme, and that's going to, of course, alter its activity. But it's has its biggest impact for those enzymes that use charged amino acids at their active site. Okay, so amino acids that have side chains that can ionize near physiological pH, those are going to be most sensitive to changes in pH with respect to their velocity. Okay, so an example of this is alcohol dehydrogenase. Alcohol dehydrogenase is one of the enzymes that converts ethanol into acetaldehyde when we consume ethanol. So we've got ethanol over here. This alcohol is converted into an aldehyde. The electrons on this carbon then go to NAD+ to reduce it to NADH. So the active site is shown in a cartoon fashion here. So this is the alcohol substrate. This is the important part of the redox cofactor, NAD+. And so the reaction is dependent on a histidine being charged and able to abstract, so the histidine pulls the OH from a serine, the OH from the serine then pulls the OH from the alcohol and allows the electrons to be taken up by the NAD+, and then that's the reaction. Then you have acetaldehyde and then reduced NADH. Okay, so if we recall, histidine is one of those charged amino acids, and it has a nitrogen on the ring on the side chain that can either be protonated and positively charged or deprotonated and neutrally charged. So that then, and that histidine is right here. Okay, so if you don't have that positive charge there and that extra proton, you're not going to get that ionic pulling of the serine, and that's necessary for the transfer of the electrons off of the alcohol to make the aldehyde. So if we were to look at velocity, enzyme velocity at different pHs, and this is looking at the alcohol dehydrogenase enzyme with this is another alcohol-containing substrate that can be converted into an aldehyde by the liver alcohol dehydrogenase. So you can see that the enzyme activity, the enzyme velocity, is peaking near neutral pH, and then as you increase the pH and have more of that histidine being deprotonated, you're going to rapidly lose your enzyme velocity. Okay, is that clear? So the way you would measure this, you would have a different Michaelis-Menten experiments conducted at different pHs to derive those data. So this is an example specific for alcohol dehydrogenase. Generally for most enzymes, they're going to have the peak activity near a place that's very convenient for our bodies. Okay, is that pretty clear? Okay, let's do a little clicker question. Is this kind of a difficult question? Okay, so select your best answer in the next 10 seconds, and then we'll try it in a different way. Okay. What if we have this diagram? So why don't you take a moment and discuss with your neighbor or neighbors, and let's see if we can change the distribution of those answers a little bit for this question.
I said 86, right? Because it's like that hasn't much to shoot up, right? Yeah. Also, I also just thought about it like. We were just talking about a lot of yeah, so we have a lot of like both are now working, but still I yes, and that like it's not to be ignored that glucokinase is also now working, but it's still not hot like I think that's what I'm gonna say. Okay. I guess that makes sense. Yeah. I think not like 6,008 is the Vmax kind of so in one place. So yeah, glucokinase is that like it's worth noting that at that concentration now glucokinase is kind of like entering the.
Okay, is this making sense? Why don't we click our best answer here and. And there's some subtle detail about this question that might hang you up a little bit, but let's see where we're at here. So I think red blood cells is the best answer. Okay, and to look at this question, let's just look at it really simply here. For this, whatever 40 or 36-year-old male, what's the substrate concentration? 4.5 millimolar, right? So where is that here? We've got about 25% of the glucokinase activity, and we're near 100% of the hexokinase activity. Does that make sense? Okay, now this is expressed as velocity as a fraction of the maximum velocity, and they may not have the same maximum velocities. So I could, if I were to ask this in a more precise way, I could say, which enzyme is phosphorylating glucose at a rate more closely or closer to its maximum velocity, or something like that. That would be a maybe more precise way to say that question. But essentially, okay, if you're fasted, you haven't had a carbohydrate meal in a few days, you need to preserve glucose for your red blood cells, right? Red blood cells don't have a mitochondria, so they're dependent on glycolysis. Okay, so this is one of those mechanisms by which the liver, its job is to pump glucose out to the rest of the body in conditions of starvation and fasting. Right, and if the role of the liver is to pump glucose out, then we don't want to be doing glycolysis within the liver, right? Because that's energetically doesn't make sense. Okay, so the liver in these situations is switching to different substrates like fat and amino acids, and thus preserving the glucose for those cells that are absolutely dependent on it, the red blood cells. Good? Okay. Okay, so this is an example of using Michaelis-Menten data to compare different isoforms of an enzyme that perform the same reaction. Okay, you could also use a similar strategy, like if you identified mutants in hexokinase that had one or more amino acid substitutions at a position, you could compare the effects of amino acids at different positions on enzyme kinetics. Okay, yes.
Can you explain how you would think about it? Like in the first question, we didn't have a graph. So can you explain how you would process that question like that?
The substrate concentration here is really near the Km for glucokinase, but the substrate concentration is exceeding the Km for hexokinase by several or orders of magnitude, right? So that's how I would approach that. Yep. Does that make sense? Or yeah, do you have a question?
Yeah, I was going to ask on the chart, what's the S0.5?
Yeah, that's a subtlety of differences between monomeric enzymes and multimeric enzymes. And meric, we know what that means. Like, if it's one protein chain that's making the functional enzyme, it's a monomeric enzyme. And if more than one protein chain comes together to make the functional enzyme, it's a multimeric. Okay, so that would be an example of quaternary structure, right? Okay, so enzymes that have more than one subunit functionally, they generally don't fit the strict Michaelis-Menten kinetics, because a modification or a substrate binding to one subunit affects the binding of the other subunit. Okay, so you can measure a Km, but they frequently refer to it as an S.5, or the substrate concentration at which you get half of the activity, but it's not a hyperbolic curve, so they don't call it the Michaelis-Menten kinetics. The curve is more sigmoidal, and then different things that activate it will push it to be more hyperbolic. So yeah, the glucokinase doesn't follow the strict Michaelis-Menten kinetics because it's got a multiple subunits. So great question though, yeah, good observation. Yes?
So just if you had like a glucose concentration of 15 millimolar or something like that, is the glucokinase ever going to match the velocity of the
okay, in this chart, yes, it would, the way that it's represented. Because the y-axis here, This is initial velocity as a fraction of the maximum velocity. So this is like normalizing each enzyme's Vmax at 100%. Yep. So, and I.
But at 15, hexakinase is still fast.
Yes. Right. If you go way beyond the scale, then.
It's going to eventually meet up with it.
Yes. Yep. Any other questions? Okay. So comparing isoforms, this is one application of Michaelis Menten kinetics, and I've got another example of that in glycolysis towards the end if we have time. Okay, let's look at drug mechanisms. And there's going to be a little bit of overlap between what Dr. Locker talked about with occupancy theory and everything, but keep them separate, I guess. They're related concepts, but the constants and what you use them for are a little bit different. Okay. So if we plot the velocity data from a Michaelis-Menten experiment, and if it's a monomeric enzyme like hexokinase, we know the data fit a hyperbolic curve, and we can describe the shape of that curve with this formula. Hyperbola are, tough to draw, right, accurately. Compared to a straight line, anyways. So we can mathematically transform the Michaelis-Menten the Michaelis-Menten equation by plotting the double reciprocals. This is called the Lineweaver-Burke transformation, and essentially it's the 1 over everything, okay, so the reciprocal. And if we do that mathematical transformation of the data that shows the relationship between enzyme velocity and substrate concentration, instead of a hyperbola, we get a straight line. So the x-axis here, this isn't substrate concentration, it's 1 over substrate concentration. The y-axis is 1 over velocity. Okay, so you can still calculate the Vmax and the Km with the Lineweaver-Burke transformation. The Vmax is going to be the intercept of the y-axis. Okay, excuse me, the 1 over Vmax is going to be the intercept of the y-axis. Is it possible to have negative substrate concentration? That's a kind of head scratcher, right? But you can just extend the line into the negative substrate space, and the Km, or the negative 1 over Km, is where that line intersects the x-axis. Okay, is that all clear? So I'm going to discuss drug mechanisms, and we'll focus on competitive versus non-competitive inhibitors. And I think that this is where there's some overlap with Dr. Lacker's material. But remember that what we're looking at is enzymatic activity. Okay, so inhibition of an enzyme activity. Okay, and I'll do this as usual, in a cartoony fashion first, and then hopefully we'll have time for some real examples. Okay, so we have a reaction here. So we have our Pac-Man-shaped enzyme, and its substrates are A and B, and when reaction happens, they join them together to make AB. Okay, so I guess this might be like a ligation reaction, for example. Okay, so many drugs act by inhibiting an enzyme activity. Probably safe to say most drugs work that way. Some drugs look a lot like their substrate, or look a lot like an enzyme substrate. They have similar chemical structures. A lot of drugs are based on the structure of ATP because a lot of enzymes use ATP. So these drugs tend to act as competitive inhibitors. So this CI here, this is a molecule that looks like the enzyme's natural substrate. Okay, so it can bind here, but when it binds here, it doesn't have the right chemistry to convert into a complex with B. Okay, so and furthermore, it's preventing any A from binding to it. Okay, so this is a competitive inhibitor because the drug is competing for binding with one of the enzyme's natural substrates. Right, that's pretty clear. So kinetically, how could we overcome that inhibitor? Yeah, what if we increase the concentration of A? Right, so if we have a lot of drug around, then the enzyme is going to more likely bind the drug. If we have a ton of A around, then the A can outcompete the drug, right? So the implication kinetically is that in the presence of a competitive inhibitor, the enzyme is going to have the same maximum velocity as it would in the absence of the inhibitor. Okay, because remember, what's the definition of Vmax? That's the enzyme velocity at an infinite substrate concentration, right? And how big is infinity? Big. And it can always get bigger, right? So you can, if you have in the Michaelis-Menten plot, you know, that asymptote, we can go, if we want to measure infinity, we can go as far as we want, right? Eventually, even if we have an inhibitor here, eventually we're going to get to a concentration of substrate that, you know, every enzyme essentially is going to be bound to the substrate, not the inhibitor. Is that key? Now, at lower concentrations of the substrates, here there's going to be competition for binding at the enzyme active sites. Some of the enzymes are going to bind the natural substrate and make a product. Some are going to bind the inhibitor and not make product. So at the lower concentrations, this is where you see the kinetic effect. And the kinetic effect is to shift the curve to make it more slanty, I guess, more leaning, and increase the Km. So at the lower concentrations of the substrate, you have to have a higher substrate concentration to get that half Vmax. The half Vmax is the same, but because the shape of the curve is shallower, you have a higher Km. Clear? Okay, so that's how it looks on the Michaelis-Menten plot. If we look at it on the Lineweaver-Burke double reciprocal plot, the Km gets larger, so the -1 over Km is closer to the origin in the presence of a competitive inhibitor. The Vmax, the intersection of the y-axis here, that's the same. Or excuse me, the 1 over Vmax. Good? Okay, let's look at another example of a non-competitive inhibitor. So the reaction in the absence of any inhibitor is shown here. So we have a substrate, it binds into the mouth of the Pac-Man, the mouth of the Pac-Man closes, and then it cuts the substrate into 2 bits. Okay. Now if we had a competitive inhibitor, that would bind somewhere in here, like at the same site that the substrate bound. Some inhibitors don't bind right at the substrate binding site. They'll bind at a different site, and that's going to change the shape of the catalytic cleft or change the shape of the entire enzyme, but it's not going to alter the binding of the substrate, of the natural substrate. So the non-competitive inhibitor binds generally at a different site from the substrate binding site, and it's going to inhibit the enzyme activity irrespective of substrate concentration. So if you think of non-competitive inhibitors, think of binding not at the active site, except there's one class of drugs that kinetically function as non-competitive inhibitors. However, they have structures that are similar to the endogenous substrates of an enzyme, and they bind in the active site. And the difference here is that for this type of an inhibitor, this is binding at the active site, and it's making a covalent bond to the active site or some type of a chemical modification of that active site that's going to permanently inactivate the enzyme. Okay, so after, and these are referred to sometimes as suicide inhibitors. So after this reaction happens, you can have infinite substrate around, but you still won't have activity of this enzyme. Does that make sense? Okay, so the non-competitive, oh, and this is just shown another cartoon example of how this can happen. Kinetically, the non-competitive inhibitors have the effect of decreasing the Vmax. Okay, they don't affect substrate binding except the suicide inhibitors, right? But you can't just keep dumping more and more substrate on and eventually reaching the Vmax. Okay, the Vmax is the maximum potential activity of the enzyme is decreased. Okay, if we look at this on a Lineweaver-Burke plot, it's the opposite of the competitive inhibitor. The 1 over negative Km doesn't change, but the 1 over the Vmax gets smaller, or excuse me, gets larger, reflecting the Vmax getting smaller.
Suicide inhibitor look the same?
The suicide inhibitor would look the same, yes. So kinetically, suicide inhibitors are non-competitive inhibitors. Yeah. Because when that active site is covalently modified, it doesn't matter how much more substrate you add onto it. It can't bind and can't react. Okay. So somebody, you had a question about the allosteric regulation and the SO5. So that's shown here as a third regulatory mechanism for enzymes, and this occurs in enzymes that are multi-subunit enzymes. So in this case, and there are examples of allosteric inhibitors and allosteric activators. So the allosteric inhibitors, the binding at one substrate, or excuse me, at one allosteric site on one subunit is going to change the shape of the molecule so that both subunits have decreased activity and decreased substrate binding. Likewise, you can also have activators that will increase affinity for substrate and increase binding. So, it's shown just in this cartoon. If you have a allosterically regulated enzyme, its activity is going to be dependent on substrate concentration like any other enzyme. But then you can maximize the activity if you have the activator around. And if we look at that in a Michaelis-Menten type of a experiment, the multimeric allosterically activated or regulated proteins don't follow that strict hyperbolic shape in with the relationship of substrate concentration to enzyme velocity. They tend to be hyperbolic, as shown here, and the SO5, that's equivalent to the Km in a monomeric enzyme. So that hyperbolic, the sigmoidal shape of the velocity substrate concentration curve for the multimeric enzymes, it becomes flatter and more shifted to the right with the allosteric inhibitors, and then the allosteric activators shift the curve to the left and make it more hyperbolic. Okay, Is that clear? Okay. Final one that I'll talk about, and this one, I'm not going to test you on it because I haven't encountered it, but sometimes I get questions about it. This is an uncompetitive inhibitor, and the uncompetitive inhibitor doesn't bind until the substrate binds. Okay, and kinetically, the uncompetitive inhibitor is both going to decrease Vmax and increase the Km. And there aren't a lot of examples of these, but the amino acid phenylalanine has been long recognized to be a uncompetitive inhibitor of the alkaline phosphatase enzyme. So shown here, this is the effect of phosphates on the alkaline phosphatase inhibitor. So what effect does phosphate have on alkaline phosphatase's activity? What's
Competitive,
okay? Good. Why do you say that?
because they have the same KM. Nope, the other one.
Same Vmax. Yep.
Thanks.
Good. Okay, so phenylalanine, if you did the same experiment in the presence and absence of the amino acid phenylalanine, this is what uncompetitive inhibition looks like. Okay, should we look at an example? Some examples here. And this is, we're going to talk about both comparing isoforms and potentially how drugs work. Okay. At the very bottom end of glycolysis is an enzyme called pyruvate kinase. And pyruvate kinase uses the phosphoenolpyruvate and ADP as substrates, and it transfers the phosphate from the pyruvate to ADP to make ATP and pyruvate. Okay, so this is at the very end of glycolysis. There are 4 different isoforms of the pyruvate kinase enzyme made by 2 different genes, and they have some different kinetic and different regulatory properties. Should we look at some data? Okay. Okay, so in this experiment, we're looking at pyruvate kinase, and let's see, yeah, I think this is the M2 isoform of it. So we incubate the enzyme, the pyruvate kinase enzyme, with a constant amount of phosphoenolpyruvate and then different concentrations of ADP. And we're measuring the rate of pyruvate and ATP formation. Okay, and we get this nice hyperbolic curve that should be very familiar to us, or should be after you get a chance to study this material for a little bit. And we can calculate then the Km for this enzyme. Now, there are 2 substrates for pyruvate kinase, right? There's phosphoenolpyruvate, there's also ADP. So we can do the reciprocal experiments where we have constant amounts of ADP and varying amounts of phosphoenolpyruvate, and we can calculate the Km for the phosphoenolpyruvate as well. So that's kind of one thing I guess I haven't mentioned, but the Km for one substrate may be different for the Km for another substrate because they're binding at different locations on the enzyme, right? Okay. So let's look at some data here. So this is looking at the M2 isoform of pyruvate kinase. We had constant amounts of ADP and varying amounts of phosphoenolpyruvate. And we were looking at this in control conditions and then in the presence of serine and fructose bisphosphate. Fructose 1,6-bisphosphate, excuse me. So the question is, what does the relationship of fructose 1,6-bisphosphate have with pyruvate kinase M2? this pretty simple? So the circles relative to the triangles are shifted to the, you can just remember the curve being shifted to the left is an activator. But think about what you're actually looking at. At any given substrate, you're going to have more activity in the presence of the activator, right? So that's activator. Okay, so fructose 1,6-bisphosphate, this is a endogenous substrate that we make within our bodies as a glycolytic intermediate. And it is an allosteric activator of this M2 isoform of pyruvate kinase. And that's shown in a different publication here with the Lineweaver-Burke transformation. Okay, so what's the significance of the pyruvate kinase M2 isoform? The M1 isoform is present in muscle and a lot of other tissues. And there's a splicing variant of that same gene that makes the M2 variant. The pyruvate kinase M2 is expressed in embryonic tissue. Okay, so embryonic tissue expresses pyruvate kinase M2. Then during development, M2 expression is stopped and M1 expression begins. Okay, so it has a role in normal physiology and development. Okay. This is a Western blot showing relative expressions of the M1, the normal adult isoform of pyruvate kinase M2, excuse me, M1, and then this embryonic isoform. Okay, so these are normal muscle cells. Anybody recognize any of these? What are these? Yep, so these are all cell lines that are derived from human cancers of different types. They all have switched to this embryonic form of pyruvate kinase. Okay? Make sense? And if you look in primary tumors, you get the same effect. Okay, so kinetically, the pyruvate kinase M2 is different. We have a higher KD and a higher KM. So the KD, that's dissociation constant, just measuring substrate binding, and then the KM, the Michaelis constant, that's looking at actual enzyme velocity, and they're both decreased relative to M1. Okay, so having pyruvate kinase M2 here, this is a choking off glycolysis, if you will. Does that make sense? Okay, so why would we do that? Has anybody heard that cancer is addicted to sugar? Have you ever heard that? Okay, so that's reflecting the Warburg phenomenon of cancers having higher glycolytic rates or higher rates of glucose uptake. But they also have this inefficient or slower enzyme at the bottom end of glycolysis. So what are they doing? Well, the hypothesis is cancer cells have increased amount of glucose going through this pathway, but it's slowed at the bottom end. All right. Now remember that principle of biochemical pathways. If you have a block in a pathway at one point, you're going to get an accumulation of the upstream substrates of that pathway. Okay, so you're going to get increased concentration of all of these different things. Now that is going to change the energetics of those reactions, and it's going to increase the propensity of the substrates to divert away from glycolysis into different pathways. And the different pathways are promoting what a cancer cell needs. So what does a cancer cell need? It needs ATP. Sure, it needs ATP, but that's not the hallmark feature of cancer, right? The hallmark feature of cancer is uncontrolled growth, accumulation of new mass, right? That is pathological mass that is displacing your healthy mass. Okay, so the idea with the M2 isoform of pyruvate kinase, it's choking off glycolysis and it's allowing glycolytic intermediates to divert into other pathways. For example, serine is made from the triose phosphates in glycolysis. We know that serine supports nucleotide production through the thymidine kinase pathway, remember, right? That's something that dividing cells need. Dividing cells are also going to need fatty acids for membranes, and they're going to need ribose sugar for nucleotides, right? So these intermediates in glycolysis, they can also support anabolic reactions that promote the cancer cell building new mass. Okay, so if you were going to target pyruvate kinase M2 as a cancer treatment, what type of a drug would you want? What? Okay, why do you say that?
Because you'd want to shift it so that it's going all the way through and decrease the accumulation of intermediates.
Yeah, I would agree with you. I'd say you would want a drug that's going to make the M2 isoform more like the M1 isoform. Okay, so that approach has been tried here. So this is a diaryl sulfonamide drug that is, one idea for targeting pyruvate kinase M2 for cancer treatments, and to try to ameliorate this block and have glycolysis going to glycolysis, not glycolysis going to anabolic processes. Okay, and I guess we have time here. Why don't we answer a couple of questions? And go ahead and chat with your neighbor about this if that helps.
Okay, so it's like trying to instead of like holding off and allowing other stuff. But don't you want the normal?
Okay, everybody selected an answer? Okay, super. Activator. It's shifting the curve to the left. Good. Let's look at the effects, again, of pyruvate kinase M2, not with a drug, but this is looking at an endogenous substance in our body, the amino acid cysteine. And pick the best answer. It might not strictly fit this, but it's pretty close to this. And if you want to talk with your neighbor, of course. Okay, should we discuss this one in the last minute? Okay, competitive inhibitor. I like that. I mean, the competitive inhibitor, these lines don't all line up right on one another on the y-axis, but they're pretty close. So maybe their instrument was a little out of calibration, or maybe this is a multimeric enzyme and it has some, allosteric regulation. But I think that of the choices here, the non-competitive inhibitor was the best answer. Okay. Okay, so it's probably not going to be a surprise to you, but you will have questions like this on the exam. Okay, yes?
Did you mean competitive or non-competitive?
Non-competitive? I meant competitive. So the Vmax is unchanged. The Km is, the 1 over negative Km is getting shifted towards the origin. So that would fit the definition of the competitive inhibitor. Yeah, did I say non-competitive? Okay, I apologize for that. You got the right answer, though. Okay, so, yes?
You said that it was important to keep this separate from the locker content. Is there a particular reason? Because there's quite a bit of overlap. But
I think there is overlap. I guess we're looking at product formation of particular enzymes, where I think the end point of the things that she's talking about are a little more context dependent. Am I wrong on that? Or, like, I mean, when she's talking about insulin binding to the insulin receptor, she's not you're not measuring the actual tyrosine kinase activity of the receptor. You're looking at like cell activation or some bigger property, right? Or some physiological response. Okay, so I think there is a lot of overlap. This is just maybe, a layer deeper looking at molecules rather than physiological systems. I think that's fair to say that. Okay, any questions? Let's see. Oh, there's 2 slides if you're interested in entropy versus enthalpy. There's 2 kind of classic examples shown here that I've given you for your own edification. You don't need that to be a physician. I'll put these quiz slides up onto Canvas shortly, as usual. Okay, bye everybody.
I was like a little like, Are you not just simply ignoring? Yeah. That would make me go like, Not to say most of that wasn't like sex. Yes. I remember. Oh. 34. Cool. When I go if there's any cornbread left,
But don't they also need?
They need energy. But I think they're have the normal ones. They just put preference towards shifting their reaction. Yeah.
Maybe they want those building blocks to get energy other ways too.
Yeah, I think they can because we're pretty efficient at making energy, but usually they're not as good as the building block. I mean, we're still pretty good at it, but relative to. Yeah, I think it's in here. It's in here too. But
You would have to have a different to be able to.
I don't know, just like right now.
There's fun.
But it said there was a notice on it.
Talking about
In 3 hours. I am, but I tried
to activate it.
So it was relaxing. It's like in a place with no windows. I didn't set it up. It was whoever did it. No, Dr. Skildum.
What is it?
You got the same thing.
Oh, the webcam? Oh, sweet.
You actually know what the weather's like outside?
My old roommate Scott works down at the museum. So if you tune into that, you can set it.
Oh, it's
I am.
1 over KM.
Yeah.
You need that
I don't think so.
Bye-bye webcam temporarily.
You're not. That's intense. It was Sunny and I got
Because one was in.